In this section, we're going to connect several core ideas that explain how cells stay alive and function. We will start by looking at energy and how it flows through living systems because nothing in biology happens without energy. From there, we will examine how cells capture energy from food and transfer into usable forms that power cellular work. We will also explore enzymes, which are biological catalysts that make life's chemistry fast and efficient. Finally, we will focus on the cell membrane and how cells regulate what enters and leaves. By the end of this section, you should see how energy use, chemical reactions, and membrane transport all are tightly linked to keeping cells organized, responsive, and alive. Energy is one of those words we use all the time, but in biology, it has a very precise meaning. Energy is defined as the capacity to cause change. Anytime something moves, grows, or undergoes a chemical reaction, energy is involved. In living systems, energy is constantly being converted from one form to another. Some energy is used to perform work, like moving molecules, building complex structures, or maintaining organization inside the cell. We generally describe energy in two main forms. Kinetic energy is the energy of motion, while potential energy is stored energy and depends on an object's position or structure. Life depends on these energy transformations happening continuously and efficiently because without energy flowing in changing forms, biological systems simply could not function. One of the most important ideas in science is the conservation of energy. This principle tells us that energy cannot be created nor destroyed, it can only be converted from one form to another. That means every biological process, from muscle contractions to thinking, depends on transforming existing energy rather than making new energy from nothing. When cells take in energy rich molecules like glucose, they are not creating energy. They're converting chemical energy into forms that can be used for cellular work. This idea helps explain why organisms most constantly acquire energy from their environment and why energy flow, not recycling, drives living systems. Whenever energy is converted from one form to another, some of it is released as heat. Heat is a type of kinetic energy associated with the random motion of atoms and molecules. No energy transformation is perfectly efficient, so heat is always produced as a byproduct. This leads us to the concept of entropy, which is measured or of disorder or randomness in the system. Every energy conversion increases entropy, meaning systems naturally tend towards disorder. Living organisms appear highly ordered, but they maintain that order by constantly using energy and releasing heat to its surroundings. In other words, life locally resists disorder while contributing to greater disorders in the universe. Another major form of potential energy is chemical energy. This type of energy is stored in the arrangements of atoms within molecules. The food we eat, the gasoline in the car, and other fuels are all contain chemical energy locked in their chemical bonds. When these bonds are broken during chemical reactions, energy is released and can be used to do work. In biological systems, cells rely heavily on chemical energy to maintain structures, grow, repair damage, and reproduce. At the basic level, life is really a carefully controlled series of chemical reactions that capture, store, and release chemical energy at just the right moment. It is useful to compare how energy transformation happens in machines and in living systems. In a car, chemical energy stored in the gasoline is converted into kinetic energy that moves the vehicle, with a significant amount lost as heat. Cells do something very similar. They take in chemical energy stored in food molecules and convert it into usable forms that power cellular work. While the mechanisms are different, the underlying principle is the same. Energy is transferred, not created, and some of it is always released as heat. In cells, the main process that releases chemical energy from food is cellular respiration. During cellular respiration, fuel molecules such as glucose are broken down in controlled series of reactions. The energy release is captured and stored in the form of cells that can actually use. Humans convert only about 34% of its energy in food into useful work. The remaining energy is released as heat, which helps maintain body's temperatures. This inefficiency is not a flaw, but a natural consequence of energy transformations governed by physical laws. When we talk about energy in food, we usually use the term calorie. A calorie is defined as the amount of energy required to raise the temperature of 1 gram of water by 1 degree Celsius. However, the calories listed on food labels are actually kilocalories, meaning one food calorie equals 1,000 small calories. These calories represents the chemical energy available in food. That energy fuels everything the body does, including movement, growth, repair, and maintaining internal balance. To help put calories into perspective by visually connecting food intake with energy use. As calories increase from left to right, you can see how different food compares to the amount of activity needed to burn the same amount of energy in about 30 minutes. On the lower end, foods like oranges and carrots or small pieces of bread require relatively few calories, which is roughly equivalent to energy your body uses just by sitting. As we move up, food like eggs or soda contain more energy and would require activities like walking, cycling, or swimming to burn those calories. Higher calorie foods such as chocolate, cake, pizza, or burgers store much more chemical energy, meaning the body would need more intense activities like running, racquetball, or skipping to use that energy. Inside cells, energy from food does not get used directly to power most cellular activity. Instead, that energy is transferred into molecules called ATP or adenosine triphosphate. ATP acts like the energy shell. It temporarily stores energy released from food molecules and energy exactly where it needs to go. Whether a cell is building large molecules, transporting substances, or changing shape, ATP is almost always involved. You can think of ATP as the immediate usable form of energy that keeps cellular process running smoothly and in a controlled way. ATP releases this energy through a process called phosphate transfer. When ATP loses one of its phosphate group, energy is ADP. That released energy can then be used to power cellular work. By transferring a phosphate group to another molecule, ATP can change that molecule's shape and behavior. This is how the cell drives processes like muscle contraction, active transport across membranes, and the synthesis of large biomolecules. Phosphate transfer is efficient, fast, and reversible, which makes ATP an ideal energy carrier. ATP powers three main types of cellular work. First, it supports chemical work such as building proteins and other complex molecules. Second, it enables transport work, moving ions and molecules across cell membranes against their concentration gradient. And third, it drives mechanical work, including muscle contractions and the movement of cellular structures. In each case, ATP couples energy release to a specific task, ensuring that energy is not wasted and that cellular activities remain tightly regulated. ATP is not stored in large amounts inside the cell because it is constantly being used and regenerated. As ATP is broken down into ADP, the phosphate during cellular work is quickly recycled back into ATP using energy from cellular respiration. This continuous cycle allows cells to maintain a steady supply of usable energy. In highly active cells, such as muscle cells, millions of ATP molecules can be broken down and rebuilt every second. All of the chemical reactions involved in metabolism would occur far too slower without enzymes. Metabolism refers to the sum of all chemical reactions in an organism, and enzymes are proteins that speed up these reactions without being consumed. Each enzyme is highly specific, meaning it catalyzes only one particular reaction. Cells contain thousands of different enzymes, each fine tuned to perform a specific task. For a chemical reaction to begin, a certain amount of energy must first be added. This initial input is called the activation energy. It represents the energy needed to break down existing bonds in the reactant so new bonds can form. In living systems, relying on a random energy alone would make reactions far too slow and unpredictable. Enzymes solve this problem by lowering the activation energy required for reactions to occur. They do not add energy to the system. Instead, they make it easier for reactions to happen under normal cellular conditions, allowing metabolism to proceed efficiently. Enzymes are extremely selective, and that selectivity comes from their structures. Each enzyme has a specific region called the active site, which binds to the particular reactant known as the substrate. The shape and chemical properties of the active site match the substrate closely. When the substrate enters the active site, the enzyme slightly changes its shape to improve the fit. This process is called induced fit. This interaction places stress on chemical bonds in the substrate, helping lower activation energy and speeds up the reaction. Once the reaction is complete, the products are released from the active site and the enzyme returns to its original shape. One important feature of enzymes is that they are reusable. The same enzyme can catalyze the same reaction over and over again without being consumed. Many enzymes are named after the substrates they act upon, often ending in the suffix "ase". This ability to function repeatedly makes enzymes incredibly efficient and essential for sustaining life. Enzyme activation follows a general sequence. First, the substrate binds to the enzyme's active site. The enzyme then stabilizes the transition state of the reaction, lowering the activation energy. After the reaction occurs, the products leave and the enzyme is free to bind to another substrate molecule. This cycle can happen millions of times per second in some enzymes. The key idea is that enzymes speed up the reaction without being changed themselves, making them ideal biological catalysts. Not all molecules bind to enzymes helps reactions occur. Some molecules act as enzyme inhibitors by interfering the enzyme's function. Certain inhibitors resemble the substrate and compete for access on the active site, effectively blocking it. Other inhibitors bind elsewhere to the enzyme and alter its shape. In both cases, the enzyme's ability to catalyze a reaction is reduced or stopped. Enzyme inhibitors play important roles in regulating metabolism and also in the basis for many drugs and toxins. In addition to managing energy and chemical reactions, cells must carefully control what enters and leaves the cell. This is the role of the plasma membrane. The membrane is made up of a double layer of phospholipids, often described as a phospholipid bilayer, with proteins embedded throughout. This structure creates a flexible but selective boundary. The lipid portion forms a barrier to many substances, while the proteins act as gateways, pumps, or receptors. Together, these components allow the cell to maintain internal conditions that are very different from the surrounding environment. Molecules are constantly in motion due to their kinetic energy. Diffusion is the natural tendency of molecules to spread out from areas of higher concentration to areas of lower concentration. Passive transport refers to diffusion across the membrane without the use of cellular energy. In this process, substances move down their concentration gradients, meaning they move in a direction that requires no energy input. Passive transport is essential for moving many small molecules or nonpolar molecules into and out of the cell efficiently. Some substances cannot cross the membrane easily on their own, even if diffusion would normally allow it. In these cases, cells use transport proteins that act like corridors or channels. This process is called facilitated diffusion. Even though proteins are involved, no energy is required because the substance is still moving down its concentration gradient. Facilitated diffusion allows cells to be selective about what enters and exits while taking advantage of passive transport mechanisms. Water movement across membranes deserves special attention because water is so critical to life. Osmosis is the diffusion of water across a selectively permeable membrane. Water moves in response to differences in solute concentration. A solute is any substance dissolved in a liquid, such as salt and water. Osmosis plays a major role in determining the cell's volume and shape, and even small changes in the water balance can have serious effects on cell function. When comparing two solutions, we use terms to describe their solute concentration. A hypertonic solution is a higher solute concentration. A hypotonic solution has a lower solute concentration. An isotonic solution has an equal solute concentration. These differences determine the direction water will move during osmosis. Understanding these terms helps explain why cells may swell, shrink, or remain stable depending on their surrounding environment. For animal cells, maintaining water balance is essential for survival. Because animal cells lack a rigid cell wall, they are especially sensitive to changes in their environment. In a hypotonic environment, water tends to enter the cell, which can cause swelling and potentially leading to cell rupture. In hypertonic environments, water leaves the cell, causing it to shrink. To survive these conditions, organisms rely on osmoregulation, which is the control of water balance. This regulation allows cells and tissues to function properly despite changes in their surroundings. Plant cells face similar challenges but have a different structural solution. The rigid cell wall surrounding plant cells provides support and prevents bursting when cell water enters the cell. Plant cells are healthiest in hypotonic environments where water flows into the cell and creates turgor pressure. This pressure keeps plants upright and supports overall structure. Without adequate water, plant cells lose turgor pressure, leading to wilting. Water balance is therefore directly tied to plant strength and survival. Sometimes cells need to move substances across their concentration gradient from an area of lower concentration to higher concentration. This process is called active transport and it requires energy. Typically, that energy comes from ATP. Active transport uses membrane proteins that act as pumps, forcing solutes across the gradient. This process allows cells to maintain internal conditions that vary different from their environment, which is critical for nerve signaling, muscle contraction, and overall cellular homeostasis. Not all materials move across membranes through transport proteins. Large molecules are often moved using vesicles. Exocytosis is a process by which materials are transported out of the cell. Vesicles containing substances fuse with the plasma membrane and release their contents to the environment. This mechanism is used to secrete hormones, neurotransmitters, and other important molecules. Exocytosis allows cells to communicate and interact with their environment in precise and controlled ways. Endocytosis is essentially the opposite of exocytosis. In this process, the cell takes materials from in by forming vesicles from the plasma membrane. One common form is phagocytosis, often described as cellular eating, where the cell engulfs large particles and encloses them in food vacuoles. Endocytosis plays a major role in nutrition, immune defense, and cellular maintenance. Together with exocytosis, it allows a cell to move large substances that could not otherwise cross the membrane.